Hello friends, welcome again in the second part of discussion of ICH Q8 guideline pharmaceutical development discussion. Friends, if you have not watched first part, please watch first part. So before starting video, just if you have any query, you can write to me on my mail ID vjsspharma@gmail.com. And friends, we have different playlists. So based on your interest, you can watch particular playlist. Please subscribe channel, share the channel with your friends. So let's start our video. Friends, in the first part of the video, we discussed about what is the product development, what are the elements of the product development, approaches, what are the approaches of development, what is the minimal approach and what is the enhanced approach, what is mean by the quality target product profile QTPP, what is mean by the CQA critical quality attributes, what is mean by CPP critical process parameter, how to link your material variables, your formulation variables, your process variable to the CQA, what are the different risk assessment tools are there which can be used for ranking of your variables, and what is the basic difference between the minimum and enhanced approach. So these topics we already covered in the first part of the video. Please watch that video. If you have any query, if you want some explanation, just write to me, or just write to me or give your number. I will add you in my WhatsApp group also for further discussion. Also friends, for a particular product, we identify variable, and then for the linking of the variables to the CQA, we use some risk assessment tools, and whatever the high risks are there, those those variables, for those factors, we run a DOE, a design of experiments. Nowadays, so many softwares are available. We run a DOE by using those softwares. We experiment. We define our responses. We place our experimental data, results of the data in that response. Then we analyze and we build a model. And based on that, we will set our goals. What is our requirement? And based on the goals, that software provides us design space. So design space is covered under the ICH Q8 guideline. So what design space means, what is the definition of a design space? It can be defined as per guideline as the multi-dimensional combination and interaction of input variables, example material attributes and process parameters, that have been demonstrated to provide assurance of quality. Working within the design space is not considered as a change. Movement outside the design space is considered to be a change and would normally initiate a regulatory post-approval change process. Design space is proposed by the applicant and is subject to regulatory assessment and approval. So friends, we decide our variable, we rank our variable, we run a DOE, and we set our goal requirement. And based on that, the DOE study will provide us a design space, ranges of the process parameter of variables, where we can get acceptable results. And if we are working within that range, then it is not a change, because based on our experimentation, based on the mathematical relationship which we obtained from the model built, it is already ensured that if we are working within that range, then it will produce a satisfactory data. So design space is that space where if we work, then it is assured that it will pass results, it will comply our requirement. So within, if we are working within design space, if a regulatory approves that design space, then if you are working within design space, then it is not considered as a change. So this is the meaning of the design space. So how to establish design space? As I already told, relationship and interaction between CPP and CQA, identification of those process parameter which can influence the quality of the product, multivariate analysis in order to demonstrate within which range process parameter can be varied without affecting the quality of the product. So we build a model, we already define our requirement based on our specification, based on the quality of the product required. Then we do the experimentation, we put the data, we analyze the variables, and based on that, model itself give us the design space. So this is about design space. Let's move to the next slide. So friends, what are the elements of the design space? So first, selection of variables, linkage and effect of process parameter and material attributes on the product CQA, identifying variables and their ranges with which constant quality can be achieved. So already I told that we will select the variables, and then we'll rank the variables, and based on the ranking we will design our experiment, we will define our factors, we will define our response, we will select a DOE design, we will build a model, we will analyze the data, and based on the analysis of data we will set our goals, our requirement, and we will get a design space. So then that design space we will define in the submission. One example is given in the ICH QA guideline. We'll cover that example in this video also. Then for each unit operation, based on the criticality of the unit operation, we will define design space, relationship of the design space to the scale and equipments. Every time it is not possible because of the resource constraint that we can do a process DOE or a DOE at the commercial equipment or at the scale up equipment. So if we have performed the DOE at the lab, then we can justify our design space for the commercial scale, for the scale up scale, based on the principle used for that scale up. Then one more thing, what is the proven acceptable ranges? See, some factors are like that we cannot study, like inlet temperature range. So we cannot particularly study that thing. Sometime in case of the product temperature range is also we will go for a range, we cannot maintain a particular set value. So a characterized range of a process parameter for which operation within this range while keeping other parameter constant will result in producing a material meeting relevant quality criteria. For that those parameter where there is a limitation of the process, we present our control strategy, we define our ranges based on the proven acceptable ranges. So also the design space and edge of the failure. For example, if you studied some ranges, some variability ranges, some process variable or some formulation variable, and whatever goal you set, entire ranges, whatever ranges you have studied, you are meeting your goal, then entire space will become your design space. But if you have studied wide ranges, then there is a particular part where you are getting design space. There are edges where if you go beyond that parameter, then you will not able to meet your goal. So those are the edge of the failure. So based on different case by case study, we can define our design space on we can define edge of failure, right? So that is the thing. Then control strategy. As we know, when we make the product development report, we have to tell the control strategy, how we are controlling our process, how we are controlling our raw material. So, based on the ranges studied, based on the proven acceptable ranges, we define our control strategy. So, let's move to the next slide. Friends, as we know that we can analyze our variable, we can study our variable by two ways. First is the OFAT, one factor at a time. We will keep remaining things constant, and only one factor we will change. For example, if you want to study binder addition time, so we will change only the binder addition time. Okay, now so like that one study. Second thing is that we can do a DOE, design of experiment, in which some factors we can study together. For example, for a wet granulation, we can study binder quantity, we can study the disintegrant quantity, and we can study the kneading time together. So what is the advantage of the DOE? That apart from the effect of one factor, if there is an interaction between the factor, that also we will come to know in the design of experiment. This is only the basic advantage of the design of experiments. Here in IC, this is the example I have taken from the QA guideline only. So here three things are there. Percent Y is the related substance, impurities, and two variables are there. One is the initial moisture content, second is the temperature, and one more variable is the particle size distribution. So based on the experimentation, these all 2D plots are plotted. So in this plot, what they are showing, that based on the slope, we can see there is an interaction between the percent moisture content, initial percent moisture content, and temperature. There is an interaction based on slope. There is an interaction, there is a relationship, a quadratic relationship is there in between the initial moisture content and temperature. Whereas the particle size distribution and the moisture content, or a temperature or particle size distribution, there is no relation, there is no slope in between that. So the at main at this we can easily predict by using the softwares uh, by using the software we can easily predict that. And by analyzing the graphs, by seeing the slope also, we can predict that there is a relationship, any relation in between the factors, any interaction between the factors, we will come to know through the DOE only, not this thing can be missed by the OFAT. In conclusion, what I want to say that DOE will give you a much better design space, much acceptable design space in compared to the OFAT studies. So just I will read this also. The depiction of interaction. The figure below depicts the presence or absence of interaction among three process parameter on the level of degradation product Y. The figure shows a series of two-dimensional plots showing the effect of interaction among these process parameter: initial moisture content, temperature, mean particle size of drying operation of a granulate drug product intermediate on degradation product Y. The relative slopes of the lines or curves within a plot indicates if interaction is present. In this example, initial moisture content and temperature are interacting, but initial moisture content and mean particle size are not, nor are temperature and mean particle size. So here in the guideline, based on slope, it is explained that which factors are interacting and which factors are not interacting. When we use DOE study, simply by seeing the p-value, by which factor is significant, which interaction is significant, we can go on the conclusion, right? So next, move to the next slide. So friends, when you run the DOE and you will set the goal, so based on the goal, you will get this type of graphical representation of the design space. So here, two parameters which may affect dissolution of a drug product are studied, and based on the study, the goal is the dissolution more than 80%. So this is the 3D plot, response surface plot to of the design space for getting the dissolution more than 80%. So you can see this is the your means space, design space, or in the contour plot also, this is a contour plot, 2D plot also, this is the space where you're getting more than 80% results. So in actual software, when you will do, you will, whenever you will put, whenever you will give a flag, you will get the values of the parameter 1 and parameter 2 which are studied. And in the parameter 1 and parameter, what are the values of the parameter 1 and parameter 2 in which you are getting the acceptable response, that is dissolution greater than 80% for this case. So these graphs are present in the guideline, and when you run a DOE through software also, you can get these two graphs. So response graphs of dissolution are depicted as a surface plot and a contour plot. So this is a contour plot, this is a response surface plot. Parameter 1 and 2 are the factors of the granulation operation that affect the dissolution rate of the tablet. They may be a excipient attribute, water amount, granule size, whatever. So let's move to the next slide. Friends, these are some examples of the design space given in the guideline. So, two examples are given of potential. Two examples are given of potential design space in Figure 1C. In Figure 1C, the design space is defined by a non-linear combination of parameter ranges that deliver the dissolution critical quality attribute. So this is a non-linear based on this slope, whatever the slope is there. So slope is, whenever slope is there, it is not linear, it will be a quadratic type equation. So in the figure 1C, the design space is defined by a non-linear combination of parameter ranges that deliver the dissolution critical quality attributes. In this example, design space is expressed by the response surface equation resolved at the limit of satisfactory response, that is 80% dissolution. So they set their goal that their dissolution is required more than 80%. So what are the value of the parameter 1 and parameter 2 in this range, in this wide range? If we, whatever parameter 1 and parameter 2 value if we keep, we'll get the dissolution greater than 80%. So this, above this will be the edge failure, and above it our dissolution will fail. Inside it our dissolution will pass based on the quadratic relationship, non-linear relationship between the parameter 1 parameter 2 established through the DOE study. So in this example, the design space is expressed by the response surface equation resolved at the limit of the satisfactory response, that is 80% resolution. The acceptable ranges of one parameter is dependent on the value of the other. For example, if parameter 1 has a value of the 46, then parameter 2 has a value of 0 and 1.5. From I think it will be difficult to see in the graph, but you can consider like that. If this is the parameter 1 value, and then parameter 2 value, we can directly understand from wherever we put our cursor, we will add a flag in the software, we will get the parameter 1 value and parameter 2 value, and inside this white area the resolution will be more than 80%. If we go means outside this white area, then resolution will fail, that it will be not it will be less than 80%. The approach in figure 1c, this is the figure 1c, means non-linear relationship, allows the maximum range of operation to achieve the desired resolution rate. In figure 1d, the design space defined a smaller range based on the linear combination of parameter. This is the linear combination of parameter. Parameter 1 has the range of 44 and 53, parameter 2 has a range of 0 to 1.1. Just they add a flag and they are telling what is the parameter 1 value and parameter 2 value. While the approach in the figure 1D is more limiting, the applicant may prefer it for the operation simplicity. See, the relationship between our response and our factors may be linear or may be a quadratic, right? So linear can be achieved, linear will provide simplicity, but it will get a compromised design space. The quadratic, we require softwares to analyze it, but it will provide a maximum use of the design space. So same is given in this example. Let's move to the next slide. So friends, one more example of design space is there where we have a two response. We have two goals. First goal is our dissolution. It should be greater than 80%. And second goal is our friability of tablet should also be in the acceptable range. So in this example, they have taken not more than means the friability should be not more than 2%. So based on that example only. So when we have two response, two goals, then design space will come like this. For example, this is the design space is by varying the parameter 1 and parameter 2, this is the design space for the resolution. By varying parameter 1 and parameter 2, this is the design space for the friability. So within this range, if we keep our parameter 1 parameter 2, we are getting acceptable friability based on the analysis of the model. Within this range we are getting acceptable resolution, but our final design space will be like that. So proposed design space comprised of the overlap region of the ranges of friability and or resolution, whatever target we take. See, this is just an example. So whatever DOE you studied, whatever parameters you studied, whatever response you defined, whatever target you kept for those response, based on that overlap design space will be the final design space. So here in this white area, you will if you will keep the parameter 1 and parameter 2, you will get both responses satisfied. I mean resolution greater than 80% and friability as per as defined by this example, acceptable range you will get as per this ICH-squared example. So similar we can apply in our projects also. So our final design space based on the response selected is like this only, a overlap design space. And this is one more example where given in the guideline. So here they are showing the upper space of the upper limit of the design space and lower limit of the design space. So in this case they are drying. So drying a particular product, drying operation is there, and upon the path, this is the path of the drying path, and the end point of the moisture content is 1-2%. And the two examples are there. If you are operating the above the design space, then excessive impurity is forming. If you are operating below the design space, then excessive particle attrition is forming. So based on the study, based on the model study, based on the experiments, this design space was defined, and these are upper and lower limits of the design space. If you work lower, then excessive attrition will be occur. If you work above, then impurity will be occur. So based on that, if any if you already defined the design space, and if you run your commercial batches, correct, and if you observe in particular batch some untoward things like more impurity or more fines, then based on your this development study, based on your design space, you can identify that how that means what is the reason of that failure, right? And same you can easily explain to the regulator also, and based on that you can propose your kappa. So these are the advantages of the this enhanced development, this scientific approach of development. So let's move to the next slide. So friends, whenever we develop a product, we will make a product development report, and in the development report also we will define our control strategy. So it is important things as per guideline. What is the definition of the control strategy? A planned set of controls derived from current product and process understanding that assures process performance and product quality. The controls can include parameters and attributes related to drug substance and drug product material and components, facility and equipment, operating condition, in process control, finished product specification, and associated methods and frequency of monitoring and control as per ISHQ10. So control strategy you will generally present in the end of the PDR. So what is a control strategy? You will analyze everything. You will, from the raw material attributes, from API material attributes, from excipient material attributes, all process parameters, all ranges, equipments, everything you will present, and whatever your understanding throughout the product development. So based on that understanding, how you arrived on the particular ranges and how we are controlling that attribute, that parameter, that ranges. So that we will define in our control strategy. Control strategy ensures that how we are ensuring that our products will be robust, our final product will meet its quality requirement, and how, based on our development understanding, how we built quality in our product, that can be demonstrated, that presented through the control strategy. So whatever development approach you follow, minimal or enhance, control strategy is always needed. And there is a difference between the control strategy and batch release. See, batch release is batch specific, but control strategy is for whole product. Each product have a different control strategy based on the development experience. Batch release involves an independent review of the batch conformance to predefined criteria through review of testing results and manufacturing records. So batch release is just the FP release, but control strategy defined for product. It is showing the understanding of the product. Has to be established based on the formulation, manufacturing, development. So control strategy at the end of the PDR will present, and control strategy at the end of development will present based on the understanding gained throughout the development. Introduced to in manufacturing, in process controls and control of drug product. So this is about the control strategy. Let's move to next slide. So friends, in the ICH-Q9, regarding the one, this example is given for the setting of specification and shown as a new shift in the approach. So now regulatory agencies wants clinically relevant specification. They are talking about the clinically relevant specification. They are talking about no end product testing, means they are talking about implementation of the quality within the process. They are talking about implementation of the PAT tools, so that batch instant batch release, we can ensure quality without even testing the finished product. So in intermediate step, they are asking for to control the quality. So this is just one example, that if there are intermediates, and one intermediate, there is a suspected that it is a genotoxic in nature, then need not to wait. We should not test the this genotoxic impurity in the final drug substance. In intermediate itself, we built some limit, like this, the TTD limit, like below toxicological threshold. That limit will control within the intermediate. For example, this compound 3 is a genotoxic, then in compound 4 only this is detected that it should be less than 10 ppm, and 10 ppm is the TTD limit, that no toxicological effect observed. So before testing the final product, in intermediate step only, it is ensured that this impurity is not present. So this example is given. So drug substance identification of the process parameters, CQP, in the synthesis influencing the generation of a specific impurity in the final product. Introduction of a specific in process control, tightening of pH range. So just example they are giving that how you can control in intermediate step the this type of impurity. So introduction of a specific purification step, these are some examples that how we are controlling the this genotoxic impurity in the intermediate itself. So this is the new shift in the paradigm of the regulatory requirements, that you should ensure quality in the intermediate itself for both drug substance as well as for the drug product. So friends, guideline is talking about the real-time release testing. We discussed earlier also that to base the release of a product on product and process understanding rather than on end product testing alone. and or on the release of the batch analysis. Means agency wants our product, our process should be that much robust that that it is ensured that it will pass the quality requirement of the end product. We should not wait for the end product analysis. We should have a proper understanding of our product and our process. This implies understanding the science around the product and process, identifying the parameter critical of active excipient process influencing the quality, establishment of control strategy risk based which monitor the important parameter influencing the CQAs, gives the basis of give basis for RTR or reduced end product testing. So examples, some examples are given for dissolution release parameter, example particle size of the active. So these are the critical parameters, critical material attributes or critical process parameters were defined, and how we can control those parameters by studying during the development study and you by presenting a control strategy, and same control strategy can be implemented during the batch manufacturing. By that you can ensure the quality within the manufacturing process itself. We should there should not be a wait for the end product testing. By manufacturing process itself we can ensure the quality of the product. So friends, this is all about the ICH Q8 guideline. Please watch first part also, then watch second part also. Please like, share and subscribe the channel. It will help us to make more such videos. If you have any suggestion, just write to us. You will provide your number. I will add you in my personal WhatsApp group also. So happy learning. Good day. Thank you. Thank you very much for your time.